Okay guys, so I'm going to walk you through our lab really quick here. What we're going to do is we're going to attempt to model the process of decoding a strand of DNA. How do scientists actually figure out what the actual bases, the A's, the T's, the C's, and the G's are if we want an entire DNA sequence of an organism? You know, remember, this has been done for the entire human genome. We've done this for bacteria. We've done it for all sorts of different creatures. And it basically is the entire code of that organism. So the steps involved in this basically are, and it will read you through this here as you go and read through this. But first, we need to make more of the DNA, right? We run PCR. We increase the frequency of the DNA, so we have a lot of copies of the stuff. And then what we need to do is we need to separate the DNA strands and basically get some new strands to build on top of those and terminate at a specific base. Now the bases are the A's, the T's, the C's, and the G's. And the way that we pull that off is by utilizing specific nucleotides that are designed to terminate the replication of that strand of DNA. Okay, so I know there's a lot of words here to understand, but essentially what we're doing is we're taking all these copies of DNA and we're going to put them into, let's say, 4 different test tubes. Now I'm going to show you a little drawing that I made here. Whoop, here we go. Over to the blackboard. So we've got our DNA here, and let's just say it's a double strand, right? We've got A's and T's paired together, C's and G's paired together, so on and so forth. Remember, they're bonded together with weak hydrogen bonds. We've got 3 bonds between a C and a G, 2 between an A and a T, and so on and so forth, right? So we make all these copies, we run PCR, and then let's just say, you know, for example, we've got 4 different test tubes here. Okay, I'm oversimplifying this by a lot, by the way. I've got to have 4 because there's A bases, there's T's, there's C's, and there's G's. Okay, now all this DNA that I copied is going to go, some of it's going to go in each of these tubes. Okay, now also, in order to make more DNA, I need to add nucleotides. So to make a copy onto a complementary strand onto the strand that's in there, I need to have A's, T's, C's, and G's to build that stuff out of, right? So I'm going to put those in each of these test tubes with it, along with the enzymes that are going to build the new DNA strands. Okay, so good so far. That would make complete new copies of all the DNA, but I want it to terminate at a specific letter. I want everywhere there's an A, I want it to stop writing the code and that little fragment to come off. Okay, the next one might stop at the next A, so it's gonna be a little bit longer. The next one at the next A, a little bit longer, okay? Now that's all kind of random. The way I do that is some of the nucleotides that I put in this A batch are gonna be specifically designed to stop it when it hits an A. Okay, so the DNA will start copying off, it hits an A, and it stops. So there's a little fragment. Okay, now some of them are going to copy all the way through, some of them are going to stop at different places, and I'll have different chunks. Now along with that, in this lab, it's going to talk about how we radioactively tag the DNA. So that's so that we can expose it on the gel so it actually shows up, because this stuff is clear, right? Even when you've got a lot of it, it's pretty hard to see. So we're going to kind of tag it with a base that's also radioactive so that we know we're just hitting the A's in this test tube, say. Okay, so I'm oversimplifying all this, but let's just say, for instance, that here's my DNA code that I took over here, TAGGC. I've got TAG, looks like I added GGC. Yep, okay. It's hard to look through the camera and also at the board. So I've got this strand of DNA, and it's got bases in the test tube with it, and some of those bases are going to cause it to prematurely terminate as it's being copied. So if it's going to prematurely terminate at an A, well, A is going to pair with a T, plunk, okay, and it's going to stop. And so I'm going to have this one little A base down here, and that's the fragment of DNA. That's it, okay? So it's going to, then I'll have some other strands that'll happen to go, you know, A, T, C, C, G, T, and then whenever I hit another T over here, it's going to pair an A with that, and it will stop, and that chunk of DNA will be left in my test tube. Okay, so I'll have all these chunks in here that I do that with A's, with T's, with C's, and with G's, and then the next step is just to run the gel. So what I'll do next, come back over here, is my gel will have individual wells at the top of it, and I'll have labeled them G, A, T, and C, and then I'll inject the stuff that I did with that I prematurely terminated with short segments of A's at the end in the A well. The ends that ended with a G will go in the G well, T, and C. I stick some electricity on this bad boy, and the fragments start to migrate down. They smooth through the gel. And once it's exposed, they're going to make these nice, neat bands where all that DNA is bunched up. Well, which one's going to go the furthest? We said this over and over again. The smallest fragment is going to go the farthest. In this case, that's this T right here. It's a single base of a T down here, okay? As I go up this, I notice that one above it here migrated this G. It must have two bases on it, okay? So there's something, and then there's a G. Well, I know what that is. There's a T on it and that G. You see what I'm saying here? As I go up here to the next one, it's a T has migrated to the next point. And so this chunk right here, I could actually read it. It would be T, G, T, okay? So in the end, when this thing is done migrating, it's really easy to read. I just read from the bottom up. T, G, T, G, T, C, C, and so on and so forth. And that's the actual code of the organism. It's pretty slick how we did that. So we're going to simulate that down in lab with some big giant posters to represent our gels. And we're going to kind of migrate stuff down and fill it in, then read it off from the bottom up. But let's look at some of the rest of the lab here. It'll kind of walk you through what we're doing on this side. But the questions, here we go, are right here. I wanted to kind of walk you through some of these. So it wants you to write the DNA sequence from the gel in figure 1. Okay, that's just this first one on this first page. I already started it for you, so you're going to read it off. T, G, T, and you'll be writing that T, G, T. I would space it out. Every 3 letters is a codon, right? So as you're reading it off, T, G, T, G. Make yourself a line or something, or at least a space. G, T, right? And I'm going to keep going there, okay? This second part, part B, you're going to utilize this, okay? And the gel, the pseudo gel that we have down in lab that's a poster board. What these are, these dark letters on the outside represent the radioactive tags, okay? These strands terminate at that tag, which happens to be an A, an A, an A. The X's just represent the fact that we don't know what those bases are. There's a whole bunch of them there, but we do know what the last one is because we designed this setup to terminate at that point. So for this part of the lab, what you're going to do is you've got to cut these guys out, like so, okay? And so I'll cut out all the C's, the T's, the A's, the G's, okay? Let me just do a couple here for you to show you what it's going to look like. Okay. So once we've got all these cut out, we're going to have a gel down in lab, a fake one, like pretend the rest of these bases, the rest of these bands aren't here, and we'll put all the C's in the C well, all the T's in the T well, okay? Then we're going to apply electricity to it, and these are going to migrate down. Which one's going to go the furthest? Well, this C here has only 2 bases on it, so it's going to go actually just right first one before the end of the gel. So it's going to go like right to there. This one's got, I don't know, 1, 2, 3, 4, 5, 6, 7, 8 bases on it, with the last one being a T. So it's going to migrate down the T well like 8 bases from the end. So to be somewhere in there. And so when we're done, we'll be able to read this sequence off, okay? So the second question here is the DNA sequence that you get from reading off all these bases here, okay, and putting them on the gel and pretending to run it down. The next step is to transcribe some messenger RNA off of the DNA that we got. So this first one is part A, okay? Later on, you're going to do part B. So part A is the code from here. So don't goof this up and use this code that you just made. So you're going to take this code here. If I have a T, a G, and a T, let's just do this first part. That T is going to translate or transcribe into an A, right? Because A's pair with T's, okay? The G is going to pair with a C. The T is going to pair with another A. See how I'm doing this? I'm going to go ahead and make myself another bar so I know that I have a stop there, or that's a codon. And I'm going to continue with the next codon, okay? Now, what happens, and I don't have one up here, if I've got an A up here someplace? If there's an A up here anywhere, remember, this is messenger RNA. I can't put a T down here. It's going to have to be a U. So anywhere you've got an A up here, don't put a T down here. It's going to be a U, okay? I'm just making it up. I don't know if that's the actual code from the activity, all right? So we've done this part. The next step is to look at the messenger RNA codons. They're right here, okay? And then create the amino acid sequence that's coded by that messenger RNA. We need the table down here. This table goes with messenger RNA, not with DNA. So make sure you're looking at the transcribed messenger RNA. We're going to translate it into an amino acid sequence. So my first one says ACA. Watch this, how I do this. It's a little different than the bullseye table. A, right there. So that means that it could be anywhere in this whole row, okay? Second base was it C, okay? That means it can be anywhere in this box right here, because this is the second base. Now they're all threonines, so that doesn't, we know what it is already, but if I needed to find the last base is an A, okay? I'm gonna find this A here, and I'm gonna go across, that's gonna put it as this top one right here. So I know it's threonine, and I can abbreviate that THR. Okay, use some lowercase in there so that you don't get confused that you're talking about an amino acid. Okay, then I'll just keep going along there, and I will translate the entire thing into an amino acid sequence. So let's talk about some of the questions here. Well, how does this polypeptide differ from most proteins? Well, take a look at your sequence of DNA. It's really short. Most proteins, most polypeptides are going to be long to be functional. They're going to be much longer than the short little sequence we gave you in this lab. Okay. They're also always going to start with a start codon. Okay, and this one doesn't. They're also always going to end with a stop codon. And I think if you check on there, you're not going to see any of the sequences that actually code for stop, like they should. Okay, even they don't. The next step here is part B. So this part here is part B. We're using this sequence of DNA up here that we got from reading this off. Okay, so don't use the other one. Use the one that we just got from reading all of this code off in lab. And it's exactly the same. Create the messenger RNA. So remember, you're using U's, no T's. No T's in this. Any place you'd put a T, you gotta have a U, right? Put a U down, okay? No T's, bad T's. Then you're gonna do the same thing you did above. You're going to read off using the codon table to come up with the amino acid sequence for this other one that we got from here. Okay, it's really easy. And once again, it's gonna ask you, how are the strands of DNA you studied in this lab different from an actual gene? And once again, an actual gene is going to be a much longer sequence of bases than this tiny little bit of code that we gave you here. Okay, it's also going to have a codon that codes for stop somewhere at the end, and a codon that codes for start at the beginning. Okay, and ours, I don't think any of ours have that in them. And that's the activity. There you go. Okay, so here's the lab. This is what we did. We took all of those letters we cut out and put them in the top in the wells. All the G's went in the G, all the A's in the A, the T's in the T, the C's in the C's. And then when we ran the gel, they migrated down the shortest fragments going the farthest. So the very first one was an A. So if I'm gonna read this off, here's the code, here's the answer: A, C, G, 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 A, C, T, A, C, C, A, T, G, 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 C, C, T, T, A. And that's the code.